Good morning, everybody. My name is Dr. Britt Nicholson. I'm the Senior Vice President for Development here at MGH, and it's my pleasure to welcome you to today's program. And the topic today is the quest to stop Alzheimer's. This is actually the 11th in our series of webinars that we have entitled Answering the Call. But just a few housekeeping items before we start. First of all, no microphone or video capabilities are needed to participate. Also, at any point during the webinar, you're able to ask questions, and you can do it anonymously if you prefer through our Q&A feature, which is at the bottom of the screen. This webinar will be recorded, and you'll receive a shareable link after the event. We really do have an all-star cast today for this topic, and I'd like to give a brief introduction to each of them. The first is our Chief of Neurology, Dr. Merit Cudkowicz. Merit is the Director of the Sean Healy and AMG Center for ALS. She's the Julianne Dorn Professor of Neurology at Harvard Medical School and is also a member of the National Academy of Medicine. Joining Merit is Dr. Bradley Hyman. Brad is the John B. Penny Professor of Neurology at Harvard Medical School. Brad is also the Director of the Massachusetts Alzheimer's Disease Research Center at MGH and is the Alzheimer's Unit Director at the MGH Institute for Neurodegenerative Disease. And Brad also is a member of the National Academy of Medicine. Rudolph Tanzi is the Joseph P. and Rose F. Kennedy Professor of Neurology at Harvard Medical School. Rudy's the Vice Chair in the Department of Neurology for Research. He's Director of the Genetics and Aging Research Unit, Co-Director of the McCant Center for Brain Health, and is also Co-Director of the Mass General Institute for Neurodegenerative Diseases. And finally, Dr. Steven Arnold. Steven is Professor of Neurology at Harvard Medical School. Steven serves as Director of the Alzheimer's Clinical and Translational Research Unit, and also as the Translational Neurology Head and Managing Director of the Interdisciplinary Brain Center. So with that, it's my pleasure to hand the program off to Dr. Cudkowicz.
Thank you very much, Britt, and thank you everyone for joining us this morning. I'm so proud to have the chance to share the great work our clinicians and scientists are accomplishing in our quest to stop Alzheimer's disease. Too many of us know of somebody who has Alzheimer's or have cared for a loved one with this disease. Our goal is to change this. Alzheimer's disease is the most common form of dementia. While Alzheimer's is not a normal part of aging, age is the biggest risk factor. Disease progression varies widely from person to person, complicating efforts to make early diagnosis, to make predictions about course of the disease, and to develop effective treatment plans. The Memory Division at Mass General Hospital was established in 1982 and has since grown to over 100 clinicians, nurses, and basic science researchers all working together. Our efforts encompass a huge program of clinical care, research, and training, from genetics research to studies of molecular mechanisms of the illness, through developing new strategies for clinical trials in AI-driven eye research. We know that to cure Alzheimer's disease, we need a better understanding of the disease's genetic makeup, which can be used to find and test therapies that will make a meaningful difference in stopping the disease progression, reversing the damage, and preventing it altogether. Our speakers today illustrate our patient-centered team approach, one that integrates our scientists with our clinical care teams, ensuring that the needs of our patients are at the center of everything we do. Doctors Hyman, Tanzi, and Arnold are internationally recognized and provide leadership at the highest levels to the National Institutes of Health, as well as numerous foundations, pharmaceutical, and biotech companies. Both Dr. Hyman and Dr. Tanzi have won the Potemkin Prize, the highest award in Alzheimer's research. We can create these integrated teams thanks to the generosities of our patients, families, and friends. Philanthropic support is critical to our ability to move from idea to therapy quickly and to make a difference in the lives of our patients. It allows us to pursue high-risk, high-reward investigations that don't fit the narrow expectations of traditional funding. It encourages our brilliant clinician scientists to pursue new ideas, to explore uncharted disease pathways, and pivot rapidly to design and carry out adaptive trials. Thank you again for joining us today. And now it's my real pleasure to turn the program over to Dr. Brad Hyman.
Thank you and welcome. You joined us this morning to learn what MGH is doing to try to treat and cure Alzheimer's disease and other dementias. I'm Brad Hyman, a clinical neurologist and a neuroscientist, and I want to represent my colleagues who together make an amazing, combined, interdisciplinary, interdepartmental team that can tackle a problem as big as Alzheimer's. I want to we've taken on and to ask for your help. So what are we doing? Well, first and primarily, we take care of patients. We firmly believe that every patient is unique. Their individual circumstances differ, their exact symptoms vary, their rate of progression varies from patient to patient. As Merritt said, every one of us knows someone who has suffered from Alzheimer's disease or their families. Every single one of them is different. Dr. Teresa Gomez-Isla, the director of our Memory Disorders Clinic, is an amazing clinician scientist who continues to create new resources that can be brought to bear for every patient. Each patient and their family needs different types of support from our medical staff, from social services, and from other health professions. And that support changes over the course of the disease. The central mission of our clinical operations is to take care of every person individually. Second, the aptly named Alzheimer's Disease Research Center, which I have the privilege of leading, has followed a research cohort of individuals who are cognitively normal, have subjective complaints, something's not quite right, or are in fact impaired. These individuals generously give us their time. They often volunteer for lab tests, for neuroimaging, and ultimately even for brain autopsy in order to work with us and together learn about how each patient is unique. This center provides the infrastructure for a community of scientists, as Merritt pointed out, over 100 of us who work side by side, the clinicians, the PhDs, the healthcare professionals, representing every specialty we can imagine: neurology, radiology, psychiatry, pathology, internal medicine, bringing expertise to different parts of the problem. We're also part of a network of 30 centers across the country. In fact, we're the oldest of those centers, and of many more around the world. But our laboratories are leaders in the research that will lead to new therapies. My own lab has helped provide the underlying data that has guided several drug trials, and we continue to work on understanding why the brain's nerve cells no longer work effectively and ultimately die. In the last 2 years, we've had some remarkable breakthroughs that both demonstrate molecular mechanisms of neuronal death, and we've learned that just like people are different in the clinic, the drivers of their disease are different across individuals. Both the specific proteins that are present in different people's brains and the specific genes that put them at risk for the disease vary across individuals. And understanding this helps us make more informed predictions about an individual's course and gives us insight about potential new treatments. We're not there yet, but we're so much closer now than we were just a few years ago. Let me give you just 2 examples of how acting on this idea that every patient is unique has influenced our research. Rudolph Tanzi has been screening drugs that may have an impact against various aspects of Alzheimer's for years, using a very novel laboratory technology that I'll tell you about in a minute. My lab's been learning to take a skin sample from patients and turn them into neurons and other type of brain cells. One of our blue sky goals is to marry these two technologies so that we can screen the panel of drugs that Rudy's discovered on the specific represent the exact genes that an individual has and see what we can learn about how to treat the individual patient. Rudy and I have worked together here at MGH for over 30 years. He'll tell you more about this and his other discoveries in a minute. A second example is the unit that Steve Arnold set up when we recruited him from Penn to MGH about 5 years ago. Now, right now, to test a drug in Alzheimer's disease uses about 1,000 individuals, takes 3 to 5 years, and costs $500 million. Our dream is to turn this upside down, instead try to develop a technique that allows us to test drugs one by one patient at a time. And we've already had success in implementing these ideas while we wait for a new physical plant to be built to help us create a new venue for clinical trials. I'm delighted to say that with the support of the MGH, of the Commonwealth of Massachusetts, and of a major gift from the Challenger Foundation leading our philanthropic support, we'll cut the ribbon of the new unit before the end of the year. Steve will fill you in on the incredible opportunities that await. I'll pass the virtual baton over to Rudy, whose insight and discoveries that drive Alzheimer's risk are so critical for our ultimate success. Rudy?
Okay. Well, thanks, Brad. I hope you can hear me okay. Yes, Brad and I have worked together for over 30 years, and we still feel that our best work is yet to come, meaning when we stop this disease. And much of what we know about Alzheimer's has come from genetics. And what you may or may not know is that MGH has an incredibly rich history in human genetics. The first time a disease gene was ever found using genetic markers, the first genetic markers of the genome, now there are 70 million, the first 5 we found here. I was lucky enough 40 years ago to be working with Jim Gasella when we were little kids, trying to find genetic linkage to Huntington's disease. And around the early 80s, we also carried out the first ever genetic studies of Alzheimer's disease. And this is all in Mass General's history and the history of our Department of Neurology. And the first Alzheimer's genes that we found, in fact, I was doing my doctoral thesis at Mass General and found the first Alzheimer's gene called APP, the amyloid precursor protein, and then 2 more after that. And those first 3 genes we found were for familial early onset Alzheimer's, and they contain mutations that guarantee the disease. And these are brutal genetic mutations because, you have folks who find out when they're 30 years old that they're going to get the disease with certainty by 45 or 46 years old in some cases. It's so tough to have these discussions with a family when you're telling someone, and then that this is the case, and then she says, well, you mean that my kids in high school are not going to know me? I mean, I'm not going to know my kids in high school in another 15 years from now. So the good news is that we're using these genes, for example, the first 3 genes for amyloid. We also discovered using genome-wide association, the first Alzheimer's gene from genome-wide association that brought us to neuroinflammation, a different pathology. The first 3 genes brought us to amyloid plaques. And now that's opened up a whole bevy of drug discovery around genes like CD33 that we discovered here again at Mass General, first neuroinflammation gene for Alzheimer's. It's amazing how many breakthroughs come out of our department that set up new programs. Just recently using whole genome sequencing now. So the entire gamut of genome sequences from over 4,000 patients and families, we found 13 new genes just published this past month that bring us to how the synapse, the connections between neurons, tend to break down. So we can put these genes into buckets, those early onset genes, and ApoE says amyloid, the neuroinflammation genes like CD33, TREM2, the genes that affect synapses. And most importantly, we integrate these, we used to integrate these, and we still do, into mice. And what we've learned over time is that mice are not particularly great mouse models for humans. Go figure, mice and humans are different. And so if we really want to recapitulate this disease, we've had to create, and again, this was first done at Mass General hospital, work of Duyan Kim and our group, of creating Alzheimer's in a dish, where we take human stem cells, turn those into different brain cells. And the key is you grow them in a gel-like matrix, because the brain is like jello, right? Jiggles around in your skull. Luckily, the skull is there to keep the jiggling down. And so when you grow these brain cells in gel, they make connections, they form a little mini brain like the size of a paper punch. So we can recreate Alzheimer's disease from start to finish, what would normally take 7 decades on average, or 8 decades, in 5 weeks. Plaques, the tangles, the neuroinflammation. And what this has done is it's made drug screening of approved drugs, developing new drugs, screening natural products, anything we can get our hands on. It's made it 10 times faster and literally 100 times cheaper, with a much better model than we had to do this in mice. As a result, one of the new drugs we have, took 20 years to get there, is a drug called the GSM that actually works against those early onset familial Alzheimer mutations, at least in the dish, so that these mutations that guarantee the disease under 50, for the first time, we'll have a drug that, if it's successful, should be able to treat those patients, where if you know you have that mutation, you start taking this drug early on and offset what the mutation does, given hopefully the first chance for a person not to have a death sentence with this guaranteed type of Alzheimer mutation. So we're going into clinical trials very soon, starting with safety, and if it goes well, and Steven Arnold's involved with that safety trial directly. You'll hear more from him in a minute, but we're hoping it'll be safe and then effective. And then because we've screened this library, all the approved drugs, every natural products, now we have about 100 compounds, approved drugs, safe natural products, et cetera. And Steven and I have been discussing how we can get those into clinical trials, just like, and Merit's on our committee as well, just like Merit has the Healy Center, the platform trial of different drugs in ALS. Now we can do the same thing in Alzheimer's with the drugs we found. We've already had a success, in fact, a crossover success, where 2 of the drugs that we helped develop in this model were successful in an ALS trial that Merit oversaw in the Healy Center. And now those same 2 drugs are being used in an Alzheimer trial. We'll be hearing about those soon, and Steven Arnold's involved with that. So the whole idea is genes to model systems, and most importantly, to drug discovery and development, and then get to clinical trials and actually translate that original genetic knowledge into helping patients and stopping this disease. And preferably, as we hope, with early intervention, early detection. And finally, I'll just say at the McCann Center for Brain Health, we're trying to find those indicators of brain health so we can have a process of early intervention by detecting early before it gets too late to start treating this disease. So to hear more about how these trials are being carried out, I'll turn the baton over to Steven Arnold.
Okay, thanks so much. You know, I feel very fortunate to be in the environment that we have at MGH. You know, as Brad was mentioning, the clinicians evaluating patients in neurology and the memory disorders unit are just superb, you know, really smart diagnosticians, and so dedicated to caring for the patients. And then, you know, Brad, Rudy, and all the scientists in, I think, 30 labs at MGH are really peerless in their creativity and productivity, generating countless ideas that we're using to improve diagnosis and treatment. So I came to MGH Neurology about 5 years ago to join the team and try to build a clinical research program to accelerate clinical trials in Alzheimer's and other dementias, and you know, bring the ideas that are percolating through the neuroscience community into people so that we can really improve their health. And as I'm sure, you know, most of you know, the current medicines we have now for Alzheimer's disease have been around for more than 20 years and really don't work very well. And despite big pharma's billions of dollars in investment and huge trials of investigational new drugs, we still lack any of the magic pills that have so revolutionized many heart diseases, stroke, and some cancers. So there are a lot of reasons for these failures, and that's what we're trying to think through. I think probably the biggest is, as Brad mentioned, the differences among people. Alzheimer's and other dementias are complicated. Amyloid and tau are important, but there's also inflammation, there's oxidative stress, there's nutritional and metabolic factors that all act in these vicious cycles with amyloid and tau to cause brain degeneration. And people differ a lot in the degrees to which these various factors drive their Alzheimer's. Now, most big trials are designed to measure how a treatment works in an average typical patient. And I've been a neurologist for 30 years, and I'm still waiting to meet that typical patient. So say you have a drug that you're testing in a clinical trial and it works spectacularly well in 10 out of 100 people, but doesn't do anything or maybe even makes people worse in the other 90. So on average, the drug's a failure. And it's typically dropped. But who were those 10 in whom it worked really well? We need genetics and we need biomarkers to disentangle the biological heterogeneity of Alzheimer's and identify people who will respond. It's their cure. Another frustration in conventional clinical trials is the way that we measure whether a person responds to a medicine or not. In a typical trial, at the beginning of the trial, you measure memory and thinking in a full day of standardized testing. Then a year later, you bring them back and you do another full day of testing. And say the medicine was working during that year, but the person slept poorly the night before the last test. They blow the memory testing and you'd say that the medicine did not work for them. We can do better by taking advantage of the digital technologies that we have, the home monitoring capabilities, to really see whether a medicine or any kind of treatment really is affecting a person in their day-to-day lives. And those are some of the technologies that we're capitalizing on as well. And then the last frustration I'll mention today is that we need to open the pipeline of medicines and other treatments in clinical trials. There are, as Rudy and Brad also alluded to, there are hundreds of already approved medicines for other conditions, as well as nutritional supplements that successfully prevent or even cure Alzheimer's disease in the three-dimensional cell culture model or the mouse models. And they may work every bit as well as new chemical entities that are being developed in the pharmaceutical industry. This is called drug repurposing. So an example might be Viagra, which we use for erectile dysfunction. It's also effective in pulmonary hypertension. That's what we call drug repurposing. So for Alzheimer's disease, there are reasons to think that certain drugs that are used for rheumatoid arthritis, malaria, HIV, certain cancers, or heart disease, that they work in Alzheimer's. They work in mouse models of Alzheimer's. We already may have the cure, but don't know it. Do they work in people? So this is the space in which we're really trying to capitalize here at MGH, is to take these ideas that work in Rudy and Brad's labs and and bring them into the people that we're evaluating in the clinics at MGH. And this is where philanthropy makes all the difference, because funding these early stage clinical trials is hard to do. So over the last 5 years, the departments of neurology, psychiatry, and radiology at Mass General got together to create the Interdisciplinary Brain Center, which is based at the Charlestown Navy Yard. And the mission of the center is really to overcome the challenges that I just laid out. So a major focus of the Interdisciplinary Brain Center is clinical trials in Alzheimer's disease, but it also helps conduct other clinical research and apply some of the same way of thinking to other diseases like Parkinson's, depression, anorexia nervosa we're working with, even long COVID, which is a trial that we're starting now in collaboration with the McCants Center. So, I think in the IBC, and especially with support from the Challenger Foundation and others, we're using some very novel, personalized and biomarker clinical trial designs. One example is we're testing a common stimulant medicine to see if it helps with mild cognitive impairment. Another is an idea that Rudy brought to me to use a nutritional supplement, nicotinamide riboside, that looks really interesting. We're doing a trial for BCG, a vaccine that's been used to prevent tuberculosis, to see if it might be useful in preventing Alzheimer's disease. And then there's baricitinib, a powerful anti-inflammatory medicine that's used for rheumatoid arthritis that may work for ALS as well as Alzheimer's disease. So we've been fortunate to be able to launch these trials. And then also, as Brad mentioned, MGH has been enormously supportive and has put in a lot of funding in order to renovate a new clinical and translational research unit over at the Navy Yard campus. This will be a 27-room space for exams, for procedures, for lab processing. We're going to have a virtual reality cave for assessments. It's going to be a hub for big data and digital health. So we're going to be sending people home with Apple Watches and iPads to kind of see how their functioning works in their day-to-day lives. So, our goal really has been to re-envision clinical trials to kind of capitalize on the brilliant ideas emerging from the labs and bring these into humans and hopefully discover new medicines that are effective for Alzheimer's. So I'm sure I went over, and I'll stop here.
Well, thank you all. This is a great jumping off point for your questions, which are streaming in. You're probably not surprised that a number of questions have to do with how much of Alzheimer's disease has to do with heredity, and how much of this is sort of environmental or things that we can control. So, Rudy, let me turn to you and ask you to comment on that.
Yeah, there's a strong genetic component in Alzheimer's disease. The first genes we found, the early onset genes, have the strongest effects. So we have 3 different genes that have over 200 mutations that guarantee the disease. When inherited, lifestyle has very little to do with the picture. So luckily, those genes are only about 2% or 3% of Alzheimer's disease, but that's when it strikes under 60. So the earlier the age of onset in your family, potentially the stronger the genetics, if you see a pattern of early onset Alzheimer's disease. And like I said, we have a new drug, this GSM, that's going into trials that we hope will reverse what those terrible mutations do. For late onset, we have a major gene called ApoE, and then we have 75 other genes clustering around neuroinflammation, some around amyloid, different pathologies. ApoE and these other genes that came out of genome-wide screens predisposed to Alzheimer's, make one more susceptible. And they account for 98% of the genetics. But lifestyle does affect their ability to bring on the disease. So a healthy lifestyle regarding enough sleep and diet, exercise, a lot of things that we talk about at the McCann Center, for example, on early protection, can offset the majority of Alzheimer's genes, including the most prevalent ApoE. So that's why we really stress that the good news is that genetics plays a role in almost every case, but in most cases, lifestyle does make a difference. And so we really emphasize that in terms of educating people on how to best reduce their risk despite their genetics.
Thanks. Merritt, I'm going to give you the next question here. As Steve mentioned, the Interdisciplinary Brain Center, I think you also mentioned the Neurosciences Center here. And so this question comes in 2 parts. One, as a department chief, what challenges do you face in terms of working with other departments? And 2, how effective do you think this approach is in terms of combining other departments to look at a single disease? So,
thank you. That is a great question. First of all, I think it's incredibly effective to work across disciplines to try to conquer some of these serious neurological illnesses. It's something I've believed since I was early faculty, that to cure these really complex illnesses, we need to be very broad in the type of people we bring in together to think about it. And the more we work collaboratively, the more likely we're going to speed up the progress. So this is really the theme of our department. Between our own labs, we have a model where our labs are open bays so that people can mingle the cooler water. It really works when people can crosstalk and see what each other are doing. The lab meetings are open, and we do that both for the clinical research and the basic science research, and that's within our department and across departments. We have such strength here at Mass General between neurology, psychiatry, neuroradiology, neurosurgery, and the neurosciences. I think last time we looked, we might have about a third of the funding that Mass General brings in for research in the neurosciences. And so we're really bringing these groups more and more together. I think the biggest challenge is probably just the depth and the amount of research and how to keep sharing that information so that we even if we might not have our labs next to each other, we know what's going on in the other building. And again, I have great partners in the other chairs and the leadership team here on how to keep doing that better and better.
Thanks, Merritt. Brad, I think I'm going to turn this one to you. We've heard that as hard as researchers are working and clinician scientists are working to develop new therapies, that to date the therapies have been disappointing. So the question is, are the therapies out there worth using at all? That was one. Two, would you encourage an individual with Alzheimer's to start these therapies, early? And three, are there things that can be done outside of pharmaceuticals that seem to have a bearing on either preventing or diminishing the effects of Alzheimer's? So,
great question. Thanks, Britt. So when a patient comes to our clinics, typically and almost universally, actually, we'll prescribe one of several approved medications. As Rudy pointed out, these are medicines that have been available for several decades already, and they have a modest effect, and that's maybe being a little bit generous. They, on average, for a group of people, the group of people who take the drug do a little bit better than the group of people who doesn't take the drug. Now, as a clinician and somebody who cares for these patients, of course, a little bit of benefit is better than no benefit at all. And one advantage of these medicines is that the number of individuals who suffer any kind of side effects is very small, and the side effects are minimal. So they're safe drugs to use, and they provide a little bit of benefit on average, primarily over time on the rate of progression of disease. So people who are on the medicines tend to progress a little bit less quickly than people who aren't on the medicines. Having said that, there's obviously room for improvement, that we really want to do better. And one of the ways in which we're trying to do better, and while we're waiting for the breakthrough of the improved medications, is that lifestyle changes, support, education of the family, education of the spouses, the social services supports, all of those can make a huge difference in the life experience of the patient with Alzheimer's and of their family. So that we can do a lot of teaching, we can do a lot of support in order to really help the patient and their family survive this journey with this devastating disease.
Great, thanks. Steve, I'm going to combine several questions here. One questioner asked if you would sort of elaborate on some of the challenges in terms of working in multidisciplinary trials with other institutions, and also commenting on how easy it is to work with pharmaceuticals. And then a second question that came in was, given what seems to be sort of a spectacular success on the vaccine front with COVID, with other institutions and companies working with one another, do you think this will catapult a push towards that same sort of cooperation now in other diseases, given the success we've seen?
Well, those are great questions, and there's a lot to unpack in in each of those. I think we let's see, yeah, we collaborate and work with pharmaceutical companies in all sorts of ways. And again, another of the joys of having moved to Boston has been just that Cambridge is now the biotech capital of the world. And there's so many great people and ideas in the larger community to work with. So we work with them in various ways. Dr. Tanzi had mentioned earlier a small company that had a combination medicine. Well, they came to, the idea kind of came out of discussions with Rudy, and then this small company came to me to say, well, how do we actually design this as a clinical trial? We put together the clinical trial. We worked with Merit's, NCRI, to kind of run now a 10-site clinical trial for Alzheimer's disease in parallel with the clinical trial for ALS. So that's kind of like an example of like a give and take that we have with the, a small emerging biotech company. I think you mentioned the issue of the vaccine, and I think that, that is an interesting model. Certainly I think there's tremendous goodwill within the academic Alzheimer community and the whole network of Alzheimer's disease research centers that Brad mentioned and that Brad leads the Mass General Center for, to kind of have a network in which to implement kind of clinical trials in Alzheimer's disease. Industry, I'm not sure, everyone still has their own kind of proprietary interests to some degree. To the degree to which they would collaborate, I think that there is that potential. We have seen that before in other therapeutic initiatives where you had, four major companies all kind of having a piece of a particular drug. So I think that is a potential, but it's a little bit complex.
Okay, thanks. Brad, one of our attendees has asked, what drugs or other means are available for people with later stage Alzheimer's?
So indeed, the therapies for right now for early onset and later stage Alzheimer's are exactly the same. And this is one of the reasons why I think it's so urgent to reimagine how we develop drugs, to think about the unique aspects of each individual who has the disease. As Rudy highlighted, the genes that are involved in early onset Alzheimer's disease are very, very different than the genes that are involved in late onset Alzheimer's disease, and yet we treat people with exactly the same medicines. And that's the sort of dichotomy between what we know clinically and what we've been able to develop therapeutically, that I think it's so important now to take from those learnings and move forward. We got to do better.
Can I comment on that? I think that you have to also consider that this is a disease that begins decades before symptoms. And we really have to get into early detection, early intervention as soon as possible. We started the McCann Center for that purpose, find the indicators of disease a decade or two before the disease strikes. And I think that's the way forward. If we want to hit these pathologies like plaques and tangles, we really need to hit these early as possible. We need to bring Alzheimer's to where heart disease is. We look at cholesterol and treat heart disease decades before there might be symptoms. Same thing with diabetes. And Alzheimer's and neurodegenerative disease, we're reactive. We wait for the brain to degenerate to the point of dysfunction. This is what we want to change. And that's why we need these early indicators of brain health. And I think the combination of the McCann Center, the IVC, the Mass General Institute for Neurodegenerative Disease, we can bring all that together so the future will be more proactive, treating these diseases before it's too late.
Yeah, I couldn't agree more, Rudy. In fact, the biggest trial right now of pre-symptomatic patients is the A4 trial, which is run by our very own Risa Sperling. And so Mass General is really taking the lead, not just in the early diagnosis and early therapy, but we actually are involved in designing those trials and figuring out how you'd run a trial like that and what you can learn from it. So you're absolutely right.
So, Rudy, I'm going to stay with you for this next question. It says, are findings in Alzheimer's disease or your approach to Alzheimer's disease significant for other forms of dementia? And if so, in what ways?
Well, if you're targeting a certain pathology, like targeting amyloid, if you're targeting, for example, amyloid, what can you hear me okay? I'm just wondering. Okay. Yeah. So if you're targeting amyloid, then specifically you're going to go for Alzheimer's. But then in frontal lobe dementias, you might be targeting the tangles rather than the amyloid. So depending on how you develop your drug, amyloid, tangles, neuroinflammation, you can decide, can I use it beyond Alzheimer's on other dementias? And you have to look at whether the pathologies overlap. One pathology that overlaps all dementias, and in fact, all neurodegenerative disease, is neuroinflammation. So we're spending a lot of time now in our 3D models and trying to find drugs that either stop neuroinflammation or protect against it, like those two that worked in combination in the ALS trial. And we now have about 50 approved drugs and natural products we found that actually are very effective against neuroinflammation in the dish, and in some cases in mice. The goal now is to raise the funds to get those clinical trials going on what are safe drugs, safe supplements and natural products, and say, do they really work? Because if we can find something that stops neuroinflammation, there's a chance to use that across the board beyond dementia into across neurodegenerative diseases. So this is a big goal for us right now.
You know, I have to say right now, we've got so many great questions in the box here. We only have time for one more. We will be, again, circulating sort of answers to the other questions sort of afterwards, even though we won't get a chance to answer them on screen. And Steve, this question comes in. Can you talk about research on the relationship to sleep in Alzheimer's?
Yeah, so that's been, that is now of increasing interest. Sleep serves a lot of functions. And one of the functions that we're discovering it serves is that, especially in deeper stages of sleep, there can actually be some clearance or greater flow of getting rid of the metabolic waste products of the brain. And so what we find is that people, as we age, sleep does become more disrupted. People who have significant sleep difficulties, as well as even sleep problems like sleep apnea, have a higher rate of cognitive difficulties later on. So I think we're appreciating how important good, restful sleep is for the health of the brain. And I think that it's a very fertile area of research, and also a very fertile area for people to try to practice good sleep, what they call sleep hygiene, in order to kind of save the health of their brain as we get older.
Yeah, we're actually doing a human sleep project now at Mass General across departments with the McCann Center, where we're looking at sleep as a diagnostic for early signs of Alzheimer's, and looking at how to use sleep as a therapy as well. So probably the most important lifestyle factor, I would say, is sleep.
Yep, and one of the therapeutic trials that just came across my desk earlier this week was to use sleep EEG as the readout for a new clinical trial, and so we're really excited about the possibilities here.
Well, I think on behalf of everybody, I'd like to thank all of our panelists, I certainly learned a lot today. And thank you all for attending. And as always, you be safe out there. Thank you.
Thank you.
Thank you.